Okay, so if you're watching this video, you'll save yourself a shit ton of time. So this week we'll be doing phage complementation test. I'm not gonna define this phrase right now. Let's just take a look at the overall goal of this lab. By the way, sorry for my voice. I got the flu, so if I sound a little bit stuffy, that's why. So, okay. I'm not sure if you have gone over the concept of complementation test in your genetics lecture, but the overall question we're trying to answer in a lab is, where is the mutation? Is this mutation exactly the same as the one we already know, or this is a entirely new mutation at a different place of a gene? So let's say if you are studying fruit flies, this is the gene, and this place is the mutation we already know. Let's call them at locus A, and this mutation will give the Drosophila the phenotype of white. And if we don't have this mutation, which is the wild type, the Drosophila will just appear black. Hypothetically, one day you saw a mutant white fly and you're wondering, is this mutation the same one as the mutation A, the one we already know, or a completely new mutation at a different locus? Let's say it's here. So how do we test for that? So for fruit flies, it's pretty easy because we can simply mate them. They are diploid, so each offspring has one copy of the gene from each parent. So if we cross the fruit fly with mutation A and the one with our unknown mutation, what will the offspring be looking like? So if our mutant gene is indeed a completely new one we never know about, that means the genome will look like this. So one gene from mutant A parent and another one from our unknown mutant parent. So since they both leave a copy of functional or normal gene, this daughter Drosophila can still make functional proteins and appear like wild type, which is black. So these two genes complement each other. So like, congratulations, you found a new mutation. So what would a white daughter Drosophila mean? That means it still got one copy from mutant A, but another copy has the mutation at the same place as mutant A. So now it has no functional copies at the gene, and it can make functional protein, and it appears white. So too bad, what you found is what we already know of. Now, our experiment is about phage mutations. It's the exact same concept. Just now our wild type phage would mean it can kill bacteria. And the mutant phage cannot kill bacteria. And we have two known mutations. One is at locus A, one is at locus B. And we're trying to figure out our unknown mutation is at locus A or B. Like, we're not trying to see if the unknown is actually some new mutations we haven't discovered yet. It's just unknown to you. Another thing is that these two mutations, it doesn't matter like where exactly are they in the genome, like as long as they're in the same Drosophila, it's fine. So like, they can be on two completely different chromosomes, they can be very close together, or they could be like on the same string of genes. It doesn't matter. Okay. So how do we figure out where is our unknown mutation? So as I said, phage is not diploid. It doesn't have two copies of the gene like Drosophila does. So we'll do something a little bit more clever, which is to inject both the unknown and mutant A into the same bacteria. We're going to test it against B also. If the unknown is at locus A, it will complement with the phage with mutation B and be normal, and the bacteria will die. And it will not complement with the phage with mutation A because it doesn't have the normal copy of the gene, and the bacteria will not die. You can do the same reasoning on if the unknown is at locus B. So that's the logic. Oh, another thing, how would you know if the bacteria died or not? So in the petri dish, if there's a bacterial growth, you'll observe bacterial colonies. So basically it'll look kind of foggy over here. But if this area of bacteria is killed, you will not see colony at this spot, and this blank spot is called a plaque. So make sure you know the difference between colony and a plaque, because I used to mess that up. Okay, so in the lab, you want to put 2 phage genes into a bacteria. The way you'll be doing it is to first put our bacteria. In this case, it's called E. coli K12, and your phage with mutation A into a tube of melted agarose and pour it into the plate. By the way, when you arrive at the lab, you'll see all of your plates already have a layer of yellow agarose in it. It's just a nutrient for bacteria to grow on, and I call it the first layer agarose, and the new agarose will be prepared will be the second layer. So now we have our known mutation, and how do we put our unknown mutation in it? So we'll not be putting it in another agarose shell. Instead, we'll just pipette little droplets on our phage with unknown mutation on the gel. But of course, we need to wait for the agarose to solidify, and then we can do it. And these little droplets will be in different concentrations of the phage. And that's why you'll be doing serial dilutions for all 3 phages at the beginning. I'll talk about it in a minute. So I forgot the order, but just as an example, so this spot is the unknown mutation with concentration of 10 to the negative 1. This is 10 to the negative 2, 3, 4, 5, 6, 7. And the last one should just be blank. So you'll be dropping TSB, the tryptic soy broth, I think. You can just think of it as water since you'll be diluting with your phage with this also. So basically, this plate is a complementation test of mutation A and X are unknown. We'll be doing another plate to test for mutation B and X. So think about what does it suggest if you observe plaques on either of them. And you should know that it will be either this or this. Like it cannot be both. Like you will only observe plaque on one of them. So why do we have 6 plates according to the lab manual? Well, the other 4 are all controls. For plate 1, 2, and 3, they are positive controls to let us see basically what the plate will look like if the bacteria can be killed no matter what. As you know, either mutation can destroy the ability of phage killing bacteria. So what if we use a different bacteria that can be dead even if the phage has a mutation? So for these 3 plates, we'll be using the bacteria called E. coli B, and it is a permissive host. And E. coli K12 right here is a restrictive host, just to like clear up the terminologies. So for the agarose you'll prepare, you'll just put E. coli in it and pour it on. You don't need to like put phage in it anymore, right? And for the little droplets, plate 1 will be your X unknown phage, and plate 2 will be the phage with mutation A. By the way, it will be labeled T4R24A because we're using T4 phage. And plate 3 will be your phage with mutation B. Okay, plate 4 will be the negative control. So this is for us to see what will the plate look like if mutation works. Basically, is to create a standard condition, and that's why we'll be using the same bacteria as plate 5 and 6, so the E. coli K12. So this plate is the simplest one. For agarose, you just put E. coli K12 in it, and you drop your X phage, your phage A, your phage B, and TSB. So think about what will this look like. So if all mutation works, then none of the bacteria will die. So like there will be no plaque. And that's all the plates you'll be preparing. Remember that only plate 5 and 6 are the actual complementation tests. The other 4 are all controls, so you should be able to predict the exact outcome if everything goes well. Now, to get solutions with different phage concentration, you'll be doing a 1 to 10 serial dilution. So the 1 means your starting phage concentration, and the 10 indicates the phage concentration after dilution. So you'll be provided with one little tube of each type of phage, and you'll start doing dilution with that. So if you pipette 50 microliters of this and dilute it tenfold, you should have 500 microliters of phage plus TSB in your new tube, right? So that means you should already have 450 microliters of TSB in your new tube. And then you'll take this diluted phage concentration and dilute it by tenfold again. And you'll be doing it for 7 times. I'll let you fill these out by yourself. So for your convenience, you should probably set up the tube rack with every tube open beforehand. So you'll be doing a round of dilution for all 3 phages. So you should be having 21 empty microcentrifuge tube ready in your rack. And that's basically all about your lab. I hope I did a good job explaining. If I confuse you even more, don't hesitate to ask. Please still read the lab manual. At least go through these steps really carefully. And I'll see you next week.